In this video, we'll be talking about how proteins are formed. This process is known as protein synthesis.
Protein synthesis involves two main steps. The first step is transcription, and the second step is translation.
Transcription occurs within the nucleus of a cell.
Recall that DNA is in the form of a double helix, and genes are short segments of DNA, and each gene codes for one particular protein.
RNA polymerase unwinds the gene that codes for the required protein and then breaks the hydrogen bonds between the bases.
The strand of DNA that's involved in the coding of mRNA is called the sense strand. The non-coding strand is known as the missense strand.
Free RNA nucleotides from within the nucleoplasm move into the DNA strand in a complementary fashion.
RNA polymerase creates the covalent bonds between the nucleotides.
Before the mRNA leaves the nucleus, it's processed by enzymes called ribozymes, which excise the junk DNA from the needed DNA.
Then the mature mRNA leaves the nucleus through the nuclear pores in the nuclear membrane.
Now translation begins and takes place in the cytoplasm.
Ribosomal RNA is produced in the nucleolus of cells, where it combines with specialized proteins to create a small and a large ribosomal subunit, which leave the nucleolus and join together in the cytoplasm to form ribosomes.
The first step in translation is initiation. The mRNA gets attached to the small ribosomal subunit. The tRNA carrying the amino acid methionine binds to the start codon AUG on mRNA strand, and then the large ribosomal subunit will join the small one.
The second step in translation is elongation. The tRNA carrying amino acid lysine binds to codon AAG on the mRNA strand.
The first tRNA leaves the ribosome without methionine. Methionine covalently bonds with lysine.
AUG is the start codon and UAG is the stop codon in this mRNA strand. Note that the ribosome moved to the right to make room for the next tRNA to come.
The next tRNA carrying amino acid phenylalanine enters ribosome and binds to the codon UUU on mRNA strand.
The tRNA that was there before leaves the ribosome, and the amino acids will covalently bond together.
The last step in translation is termination. The ribosome moves to the right and the stop codon occurs on the mRNA. A stop codon is a codon for which there is no corresponding amino acid.
The release factor will come and bind to the stop codon. The tRNA is separated from the mRNA, which is separated from the small ribosomal subunit, which gets separated from the large subunit.
The protein is now made.